Hello everyone. So in continuation with the molecular biology series of lectures, today we shall look into cell cycle, briefly DNA repair mechanisms and defects of DNA repair mechanisms. So let us proceed. So DNA proofreading and repair. The mechanism used for repair of DNA involve first recognition of error in DNA. So this is a DNA fragment. This is the 5' to 3' end, and this is the 3' to 5' end. And as you can see here, that this is guanine here, and the base which is coming to pair with guanine is thymine, which is an incorrect base pairing. So the first step in correcting this or repairing this mistake is recognition of the error, which is done by DNA polymerase. The second is removal or excision of the damaged region of the DNA strands. So this is the wrong pairing, and the removal of this wrong pairing is taken care by different enzymes in prokaryotes and eukaryotes. In prokaryotes, it's DNA polymerase 3 and exonuclease, whereas in eukaryotes, it is DNA polymerase delta and epsilon, which act as exonucleases. So in prokaryotes, it's DNA polymerase 3, whereas in eukaryotes, it is polymerase delta and epsilon. Both are exonucleases. Now, once this wrong base is removed, there is a gap left or a hole, and this hole needs to be filled. So the filling of gap left by the excision of the damaged DNA is done by DNA polymerase. And now once this is excised and removed, the gap will be filled. So the right base is now attached. So you can see that guanine is attached to cytosine. And once that is attached, finally sealing the nick in the strand that has undergone repair is done by ligase. So this is how the DNA proofreading mechanism occurs, and it repairs the incorrect base pairing which occur in normal replication processes. Now we come to the first kind of repair mechanism. Let us look at the details. This is the base excision repair mechanism, where you can see that these are the two strands. This is the 5' to 3' and 3' to 5' end. There is single base alteration in base excision repair, and this single base alteration is detected by HSMUG1. This is a protein that removes any uracil residues in the single stranded DNA during replication or transcription. As you know that DNA does not have uracil, so by mistake if there is a uracil, then it is removed by the glycosylases. So let us see how it does it. So this removed. So different glycosylases remove these modified bases. The modified bases is by deamination, and for uracil it is uracil glycosylase. So once this uracil is removed, there is a apurinic site, and this is the apurinic site. And then this apurinic site will be cut. So a nick is made by endonuclease at the apurinic site, and the nearby bases are also removed from here. And then DNA polymerase will fill this gap by bringing in the correct nucleotides according to the complementary strand. And now you can see that here at the third position, where there was uracil earlier, now it is replaced by cytosine. So cytosine is pairing with guanine. So the correction has been made by base excision repair. That is, the wrong base is removed by exonuclease, and then the correct base is brought in by DNA polymerase, and it is sealed by DNA ligase. So nucleotide excision repair, it is an important mechanism by which the cell can prevent unwanted mutations by removing the vast majority of UV-induced DNA damage. Now, for damaged DNA segment up to 30 nucleotide length can be repaired by this mechanism, which is generally caused by ultraviolet light, which induces thymine dimers. It repairs covalent bonding between adjacent thymine bases. The base modifications, strand breaks, and strand linkages are caused by chemotherapeutic drugs, ionizing radiations, environmental chemicals. There are 9 major enzymes that are involved in repairing the defective segment. The names of these enzymes are based on the disease. These include XPA, B, C, D, E, F, G, and here XP stands for Xeroderma pigmentosum, whereas CS stands for cocaine syndrome, so CSA and CSB. DNA polymerase polymerizes and DNA ligase seals the gap. Nucleotide excision repair mechanism, or the NER. As you can see here, this is a thymine-thymine dimer formation. This thymine dimer formation leads to DNA damage due to the thymine dimers, and once this error is detected, then the excision of the damaged region is carried out by the DNA, and the enzyme that does this is UVR ABC exenuclease in prokaryotes. And in eukaryotes, there are a different set of enzymes which carry out the excision. Once the defective part is removed, DNA polymerase comes and synthesizes, and the DNA ligase ligates the strand. So, the NER mechanism repairs covalent bonding between adjacent thymine bases or adjacent thymine cytosine bases, which are caused by UV light. So the defects that are caused due to DNA repair or replication include Xeroderma pigmentosum. Now, Xeroderma pigmentosum is a autosomal recessive genetic disorder. Here, the XP genes, that is A to G, are present, and the UV light causes the thymine dimers. The mutations in these genes, these XP genes, in nucleotide excision repair mechanism leads to Xeroderma pigmentosum. More than 1,000-fold sensitivity to sunlight is increased. It can cause skin cancers, and death generally occurs within the second decade of life. There is inability to repair the damage of ultraviolet rays to the skin is seen in Xeroderma pigmentosum. So this is a picture of a child who has zero means dry skin and pigmentosum means pigmentation on the skin. So this is the physical manifestation of Xeroderma pigmentosum. Now, the third mechanism is the mismatch repair mechanism, in which there are different proteins that participate. Their names are mute proteins. The mute proteins include mute S, which scans the DNA and it recognizes the mismatch. So, as the name says, mismatch repair mechanism, these proteins recognize the mismatch repair. Then mute L, it links with mute S and mute H and activates the mute H. So the mute H finally binds to the hemimethylated GATC sequence. So this is how these proteins play a role in mismatch repair in identifying the damaged part. So let us see the whole mismatch repair mechanism. In mismatch repair mechanism, this is the strand, this is the parental strand which is methylated, and this is the newly synthesized strand which has no methylation. So methylation marks the older parental strand, And now you can see that guanine is pairing with thymine, which is mismatching of base pairs. And this mismatch has to be first identified. This is first the methylated strand is identified, and this methylated part has GATC complementary to which is CGAT. Because of this methylation, the parental strand can be identified from the daughter strand, and then the error can be checked. So the proteins that are involved are MUT S, H and L. The MUT S will scan the DNA by forming a clamp-like complex. The MUT SL will find unmethylated GATC sequence. MUT H will bind to the MUT L and MUT SLH complex is formed. The MUT H is a site-specific endonuclease. It cleaves the defective strand near the GATC. So this is the GT mismatch, and now the MUT S will scan and the whole DNA, and MUT SL will come and identify the unmethylated GATC sequence. And once the unmethylated sequence is identified, they will cleave the defective strand near GATC, and then additional nucleotide at the 5' to 3' end are also removed. So there is more degradation around this area. As this strand is unmethylated, this is removed, and then DNA helicase will unwind, single stranded proteins will come and bind to it, and the defective part is removed by exonuclease 1 activity. Then the DNA polymerase 3 will come and it will replace the defective bases with the uh, complementary base pairing, and DNA ligase will seal the gap. So this is how the mismatch repair mechanism works. In humans, non-polyposis colon cancer is seen. It is also abbreviated as HNPCC, and it is a autosomal dominant inheritance. It is associated with the defective gene mismatch repair gene MMH. These genes are named as HMLH1 and HMSH1 and HMSH2. The syndrome is characterized by familial predisposition to colorectal carcinoma and extracolonic cancers. It has very high penetration, around 85%. There's an early onset by the age of 42 years. The treatment is removal of almost all or most of the colon. So this is about human non-polyposis colon, which is a defective in the mismatch repair mechanism, mismatch repair. The double strand repair break mechanism. So this is the fourth type of repair mechanism which exists in our body. In this, there is exchange of genetic information between DNA segments. So let us look into what does the double strand break repair mechanism hold. Now, due to excessive exposure to UV rays and X-rays, there is some kind of double-stranded DNA break, and this can be repaired by non-homologous end joining repair mechanism, and it is the primary pathway for repair of double-stranded breaks. It is the most preferred type of repair mechanism for the G1 phase of cell cycle of the mitosis, and this mechanism relies on the Ku protein. The exchange of genetic information occurs randomly. It need not be between identical sequences. So that is why it is known as non-homologous end joining, because the joining of the chromosomes, uh, there is exchange of genetic information which is random, and so they need not be identical sequences. So that means it is non-homologous. The second mechanism is the homologous recombination repair mechanism, which is abbreviated as HRR, and it is a preferred mechanism in the S and G2 phase of the cell cycle. It facilitates error-free repair. It maintains the genetic integrity. There is exchange of genetic information between identical DNA sequences or parts of chromosomes, and this is something opposite to the NHEJ mechanism. The deficiency of homologous recombination repair is associated with several tumors like prostate, ovary, breast, lung, etc. In non-homologous end joining, the two broken ends of DNA are pieced together. So, at first, the double-stranded break is recognized by end binding proteins. So these end binding proteins have recognized the double stranded break, which is caused by high radiations. Then these proteins recognize additional protein to form cross bridge that prevents the two strands from drifting apart. So the additional proteins come and align here so that these two strands do not drift apart. Next step is addition of proteins are recruited to the region that may process the end of the broken chromosome by digesting particular DNA strands. So here these proteins align and they do the DNA processing. Once the DNA is processed and the defective parts are removed, the gaps are filled by DNA polymerase and then the DNA ends are ligated together. So this is about the steps in which non-homologous end joining repair takes place, and there are four steps. First is recognition, second is alignment of proteins, third is processing of the DNA, and fourth is gap filling by DNA polymerase and sealing by DNA ligase. Now we look at the homologous recombination repair mechanism, which is called homology directed repair, which occurs when homologous DNA strands from a sister chromatid are used to repair the double stranded break in the other sister chromatid. So these are sister chromatids and there is a break over here and the two broken chromosome pair up with its homologue and then the damaged region is replaced via recombination using sequence copied from the homologue. So let us look at the steps. At first, the double stranded break is processed by short digestion of DNA strands at the breaking site. This is followed by DNA exchange of strands between broken and unbroken sister chromatids. So this is the unbroken, this is broken. There is exchange between them and then the unbroken strands are used as templates to synthesize DNA in the region where the break has occurred. Finally, the crisscross strands are broken and then rejoined to produce a separate chromatid. So here separate chromatids are formed. So this is how the homologous recombination repair occurs. Now these are the various types of errors that we have discussed. These are the causes. That is, the errors may be due to replication, due to radiation, chemicals, reactive oxygen species, and chemotherapeutic drugs, which can cause damage to the genome. The genome then has a checkpoint activation in the cell cycle, and the cell cycle first undergoes cell cycle arrest, which means if any damage is formed, first the cell cycle stops uh, further replication of DNA. There is chromatin remodeling, DNA damage tolerance pathway begins, and the DNA damage repair mechanisms now start. If the DNA damage is not repairable, then the cell may go for cell death or apoptosis, or it may lead to cancer. So these are the different consequences of DNA damage in case the repair mechanisms are unable to repair the damage. Mutations. Mutation is defined as a permanent change in the nucleotide sequence of DNA that is transmitted in future generations of the cells. So as you saw that the DNA while replication has its own proofreading mechanism, and during the replication process if there are any errors, that is corrected by the proofreading mechanism. And during cell cycle, in the different phases of cell cycle, there are some other mechanisms which correct errors that are caused by different agents. This has already been explained in one of the other lectures titled as DNA damage and the agents causing DNA damage. Now, Ataxia telangiectasia. This is a rare neurodegenerative disease that affects many parts of the cell and it causes severe disability. Defect in gene that detects DNA damage is seen, which means that the uh, method or the mechanism by which the DNA damage is detected, that itself is defective. So the gene for detecting the damage is defective. And the ataxia telangiectasia mutation is on the gene located on 11q chromosome. The mutated gene is present only in 1% of the population. It affects the brain and other parts of the body. Ataxia refers to uncoordinated movements such as walking. Telangiectasias are enlarged blood vessels or capillaries just below the surface of the skin. So, the cerebral ataxia is there is incoordination or lack of balance. And ocular telangiectasia is widening of the small blood vessels in the conjunctiva. So, this is ataxia telangiectasia in which there is a defect in the gene that detects DNA damage. Defects in DNA repair or replication, we have seen two different repairs. That is, um, that is, we discussed about Xeroderma pigmentosum, and we discussed about Cockayne syndrome, and then we also discussed about uh, non-polyposis cancer. Now we will talk about the other defects of DNA repair and replication. So, Fanconi anemia, it is caused by gene involved in DNA repair. There is increased risk of X-rays and sensitivity to sunlight. The second defect is Bloom syndrome. It is caused by mutation in a DNA helicase gene. There is increased risk of X-ray uh, exposure and sensitivity to sunlight. Then we come to the Cockayne syndrome. It is caused by defect in transcription linked DNA repair. There is increased sensitivity to sunlight. And finally, the Werner syndrome, which is caused by mutation in DNA helicase gene, and there is premature aging. So these are the different defects in DNA repair or replication mechanisms. So let us take a quick look at the various mechanisms that repair DNA damage in our body. This is a summary. So the first kind is the mismatch repair mechanism, and it repairs base pair mismatch. So here you can see thymine is paired with guanine, whereas it should have been guanine with cytosine. So this mismatch, this kind of base pair mismatch is repaired by MMR or mismatch repair mechanism. The second type of mismatch repair mechanism is the NER or nucleotide excision repair mechanism. It repairs bulky adducts that are formed or the pyrimidine dimers that are formed. So this is a pyrimidine dimer, this is a bulky adduct. The third type of repair mechanism is the base excision repair mechanism, and it repairs A basic sites. So there are no bases here, and it repairs uracil. So guanine should have been paired with cytosine, but there is introduction of uracil here due to modification of base. So and the single strand breaks. So base excision repair mechanism repairs A basic site, uracil, and single strand breaks in the DNA. The fourth type of repair mechanism includes homologous recombinant repair mechanism and non-homologous end joining repair mechanism. Both are separate mechanisms, and they repair double strand breaks. This is a double strand break, and they repair inter-strand cross links. So this is the summary of all the different repair mechanisms and the kind of DNA damage they are capable of repairing. Thank you. Please like, comment, share and subscribe to my channel. Thank you for watching.